There are a lot of non-parametric tests out there in the world. We're just going to go over a few of the more simple ones, but know that there are quite a few of really interesting tests that you can do if you go further in statistics that don't rely on the data following the assumptions of normality. And those can be really powerful and useful. Tests. Now, these are covered in chapters, two of the tests are covered in chapters 21 and 22 of your textbook, but most of the rest of the tests are not covered in your textbook. You'll have to look them up in other sources, but I'll point you in the direction of some of those other sources. The learning objectives for this lecture are to understand the definition of non-parametric tests, look at the circumstances for the use of non-parametric tests, look at some examples of common tests and their parametric equivalents, and then delve deeper into how to use the chi-squared tests. So what are non-parametric tests? Very simply, these are methods of analyzing data that don't depend on parameters like means and variance. They're not based on theories of probability distributions like the t-tests or the ANOVA, and the model structure is determined from the data. They don't require your data to be normal. Now, when I looked up non-parametric tests, and I did right before this lecture, I spent some time revising the lecture and looking up more about it, because I always learn something new when I delve deeper. And different websites say different things about what a parametric versus a non-parametric test is. My understanding is that it really just means that it doesn't depend on parameters like means and variance, but other websites say it doesn't require your data to be normal, which, as far as I know, is also true. So these are very useful. These are very useful when our data just don't work with the other forms of analysis. Okay, when to use the non-parametric tests? Well, again, use these tests when your data do not meet the assumptions of the parametric tests. For example, when you have samples with extremely skewed data instead of normal distributions, when you have samples with extreme outliers, or non-numerical data like categorical, nominal, or ordinal data, or, well, categorical is a type of data, and then the two subcategories are nominal and ordinal. If you have ordinal data, it might look like numbers, but it's not going to follow a normal distribution. You can still analyze it. You just have to use a different test. And you can also analyze data if all you have are categorical data. You don't have any numbers. Well, you have counts, but all of your response variable and your explanatory variable are both categorical. And we'll look at how to do that. That's what we are going to focus on this week. So remember one thing: when you graph your data, if you want to compare measures of central tendency for your data, use the median instead of the mean for graphing your data, because the mean is really easily pulled off center by strong outliers. If your data are skewed, and we looked at this a lot, remember when we made a lot of different histograms and we put the mean and the median on there, and we could see that for the skewed data, the mean was pulled strongly over towards that skew, whereas the median stays pretty much in the center of the data. So it's better to graph the median than the mean when you have really skewed data. However, notice that if you look online, you might find people saying that some of these non-parametric tests actually compare the medians. Now, my understanding is that's not really true. They're not comparing the medians. It's just that the medians are the handier thing to put on the graph to make a visual comparison when you're representing your data. We'll go more into that. Okay, why not to use these? If they're so flexible, why not use them all the time? Then you wouldn't have to go to all the trouble of trying to check the assumptions of your data with all these histograms and box plots and whatnot. Well, first of all, they usually have less power, although that's not necessarily true with some of the newer statistics. But typically, yeah, they're less powerful tests. They may not compare the parameters of interest or help you to estimate the parameters of interest as well, because you're not going to get, say, a mean and standard error or confidence intervals and be able to compare multiple groups in the same way that we were able to do with the ANOVA and the two keys test. Remember, with the two keys test, we could look at how each group differed and we had all those great confidence intervals. We'll still get p-values for comparing two groups, but we won't be able to break it up in the same way. Also, some of the more complex analysis you might want to do in the future are trickier to do with a non-parametric test. So the rule of thumb is, if you can do a parametric test, do the parametric test. In fact, I have seen a paper where it suggested that doing the non-parametric test instead of the parametric test was a way of kind of hiding the truth of your data and was considered unethical for medical studies. Oh yeah, and it's nice to know that because of our central limit theorem, even when the data don't quite meet the assumptions of normality, if the sample size is large enough, we can usually use the parametric test anyway. But I'll show you some of these non-parametric tests so that you'll know what to do if you need to use them. We won't go into them a whole lot. So here are a group of non-parametric tests that you'll probably run across at some point. There's the Wilcoxon signed rank test. They all have horrible names. They have horrible names, and a lot of them have multiple names. It's hard for me to keep track of them, but I'll show you a table on the next slide that sort of breaks it down. So this is a non-parametric version of the one sample t-test, or the paired samples t-test. And we looked at this when we did the t-test lab real quick. We looked at how to do the Wilcoxon signed rank test for the paired t-test. So if you want to do a one-way t-test or a paired t-test and your data don't meet the assumptions, you can do the Wilcoxon signed rank test. And it's set up almost the same way. It's Wilcoxon test, and then you put in your XY data and it spits out a W statistic and a p-value. And you can use this for ordinal data, like the number of stars of a hotel, right? It's there, that's a number, but it's an order. It's not really a numeric variable in the same sense as the continuous variables that you would measure. So that would be an appropriate data type to use for the Wilcoxon signed rank test. Then there's the Mann-Whitney U-test, also called the Wilcoxon rank sum test. So there's Wilcoxon signed rank test, Wilcoxon rank sum test, which is the same as the Mann-Whitney U-test. I told you the names were horrible. So this is a non-parametric version of the independent samples t-test. So if you have two independent samples for your t-test, then you use this test. And the test primarily deals with two independent samples that have ordinal data, or it could just be really small sample sizes or really skewed data. And then there's the Kruskal-Wallis test. And yeah, there's probably a bunch of history of some people named Kruskal and Wallis and Mann and Whitney and Wilcoxon. You can look them up if you're interested. You know, some of these historical figures have colorful stories. This is a non-parametric alternative to the one-way ANOVA, which we learned about last week. It's used to compare more than two independent groups with ordinal data, or again with, you know, heavily skewed data or data with outliers. And finally, there's the chi-square test, which is used to test data where you have one or two categorical variables and you don't have a numeric response variable at all, right? Here's a handy dandy table. And yeah, I'm posting this lecture onto Laulima, so you'll be able to see it. And this breaks it down for you. If your data setup, look, if you've got one variable with two categories and you're comparing between subjects, the parametric test would be the independent t-test, and the non-parametric equivalent is the Mann-Whitney U-test. If you have one variable with two categories within subjects, like our jumping jacks, one variable was pulse, two categories before and after within subjects measuring the same person twice, the parametric test is the paired t-test. And the non-parametric test is the Wilcoxon signed rank test. If you have one variable with more than 2 categories and you are comparing between subjects, then you use the one-way ANOVA for parametric data or Kruskal-Wallis test for the non-parametric test. And if you have one variable with more than 2 categories within subjects, that would be the repeated measures ANOVA, which we have not covered yet. And you could use either the Friedman test or the Moods median test. And then there's the one variable correlation, and you could either use the Pearson's or the Spearman's row. Is part of this slide covered up by anything on the screen? I know I've got my little talkie box now. Okay, you can see it good. So memorize, memorize this table, especially, no, I'll tell you now, I am not going to require you to know the non-parametric equivalents of the parametric tests. I'm not, because frankly, if I have to use them, I Google it. I say, Google, what is the non-parametric equivalent of the one-way ANOVA? And then I run it. And to be honest, these tests they're great, they're useful, they work, they're totally valid, but truth be told, nowadays there are more modern alternatives that if you're going further in statistics, you would probably want to learn. But these are nice. I mean, they're very simple. We could easily calculate them. And in years past, professors have had worksheets for you to calculate them. I'm not going to have you calculate them. Only the chi-square I'll have you calculate because I want you to really wrap your head around it. But just know that these are actually really simple tests to do computationally. The assigned rank test, you're really just ranking all the values and then putting a sign like here's number 1, here's number 2, here's number 3. And then you rank them and you compare those, you sum them up and compare the ranks. It's really, really simple mathematically and really handy in that way too. Okay, so we've got all these different kinds of tests. This excludes on this table the chi-squared test, which is what we're going to talk mostly about today. Okay, the chi-squared test, required reading for this week, chapters 21 and 22. I know 2 chapters, that's a lot, but they're fairly short and they're actually really interesting. They go over a lot of really good examples, and that's going to help you so much to understand what's happening. I can explain only so much during the lecture. Spending your time delving into the textbook is going to give you a much deeper understanding of these tests. Overall outline here for the rest of the lecture, we're going to look at these 2 different types of t-tests. 2 types of t-tests, or 2 types of chi-squared tests. We had 3 types of t-tests, and we have 2 types of chi-squared tests. One type of chi-squared test is the one-way, you just have one categorical variable. The other type is the two-way, you have 2 categorical variables. Pretty simple, kind of like the 1 sample t-test and the 2 sample t-test, right? We're going to go over the hypotheses for these tests, look at how you calculate the expected counts and the chi-squared statistics. We're going to look at interpreting the significant chi-squared results and conditions for the chi-squared test. Okay, chi-squared conditions. Yeah, apparently not in that order. The chi-squared test. So for both of these tests, you want to make sure that your observations are randomly sampled from the population of interest, right? And that removes bias from the sample. As long as you know what your population of interest is, and that's the population about which you want to make inferences, right? What do you want to talk about? You want to talk about students? You want to talk about students at UH? You want to talk about students in the United States? If you want to talk about students at UH, you want to randomly sample from all the students at UH. You want to talk about students in the United States, you have to randomly sample from all the students in the United States. That way you can make inferences about that population from your sample. So make sure you've randomly sampled from the population of interest. Make sure your observations are independent. That means one observation does not affect the other observation. And each observation falls into only one category, and that you have an adequate sample size, and I'll go into more detail of that on the next slide. So there's 2 types of tests, as I mentioned. There's the chi-squared goodness of fit test. And again, this is when the explanatory variable is categorical. It's a simple random sample with any number of levels. And the response variable has known frequencies. You can expect what the frequency is going to be. And in our lab on Thursday, we're going to use M&Ms. So our explanatory or categorical variable is color of the M&M with how many different levels? There's blue, brown, green, red, orange, and yellow, right? We've got 6 different colors. So those are the number of levels. And the known frequency, well, we expect in this case that the number of that the frequency is evenly distributed. So we should get an equal number of blues as browns, as yellows, as reds, etc., so that each one of them should be 1/6 of the population. So our known frequency is 1/6. And then there's the chi-squared test for two-way tables. That's when you have 2 different variables. It's also sometimes called the test for independence or the test for association. And this is when your explanatory variable is also categorical, and it can have any number of levels. And your response variable is categorical. It can have any number of levels. And for your homework, you're going to do this test looking at males versus females in the class in this semester versus last semester. So we have 2 categorical variables, male and female, or sex, and what semester. Those are the 2 categorical variables. And we're counting the number of students that fall into each category. Now, again, each observation can only fall into one category. If a student took the class in both semesters, that would be awkward. It wouldn't fit the assumptions anymore. Fortunately, that hasn't happened. Everybody passed. Or if someone had 2 genders, which, could happen, but it didn't happen this time. So we didn't have to worry about that. So it meets the assumptions and adequate sample size. And again, I'll go into that right next, I think yeah, here we go, sample size. Okay, there's a rule of thumb straight from the textbook. We can use the chi-squared test when no more than 20% of the expected counts are less than 5. That means how many you expect to be there are less than 5. So you have to calculate your totals, multiply that by the proportion that you expect, and then make sure that those should all be 5 or more. And all the actual observed counts are at least 1. A 0 here and there won't completely kill you, but it usually will cause a problem. So if you do a chi-square test and you've got zeros in there or really small numbers, you'll get an error message. You'll be like, why? That's why, right? You need a certain sample size. Now, one way to handle this is that you can collapse your categories. Like, let's say we were counting up our M&Ms and I only had a couple packs of M&Ms and I didn't have enough. I only got like 1 green one or 0 green ones. I got 0 green ones. I could say, well, maybe I'll just put like blue and green together in one category, and I'll put brown and blue in one category, and I'll put orange and yellow in one category. I don't know. Do something like that, right? Wait, I forget. Which was brown and oh no, you'd have to put red and brown together. Yeah. So I was like, where's red going there? Right. You could collapse the categories and that would increase your counts per larger category, but you lose information when you do that, right? You lose information about the detail of what's happening with the colors. So it's better to just collect more data if you can go get some more M&Ms. They are anyway delicious. All right, more about this goodness of fit chi-squared test. Again, this is a little bit of a hard name, goodness of fit test, but it sort of makes sense because we're looking at how well our data fit the expected proportions, the expected probabilities or the expected frequencies, right? So we're seeing how well our data fit. So the goodness of fit, do they fit real good or not? And if not, then we'll have a significant difference and we'll reject the null hypothesis, right? And if you think about it like this, here's the example from the book. If you you took a random sample from a bunch of hospitals and you counted up the number of births on each day of the week because you wanted to see, is the number of births on each day of the week the same? Or are babies more likely to be born on one day of the week than another? You might have some reason for thinking that, you might not, but you just want to find out. So you can take your observed sample proportion, which is a simple random sample, in this case of 700 births, and you can see, okay, well, we've got 15% on Monday, something like 18% on Tuesday, right? You could graph it up. And then you can look at, here would be your expected proportions under the null hypothesis, which is that the proportion for Monday is equal to the proportion for Tuesday, and so on, and so on, and so on, which should be equal to 1/7, right? If they were all the same, then you would have 1/7 of the births on Sunday, 1/7 on Monday, 1/7 on Tuesday. And since you have a sample of 700, that means you would have 100 births on Monday, 100 on Tuesday, 100 on Wednesday. And you might have some variation from that, but then you would look at your chi-squared statistic and see if that little bit of variation is just little enough to be expected under a random chance, or if this is really a significant departure that would tell us, hey, something's going on. Babies are more likely to be born on, well, what day do you think, looking at the graphs? It's right here. It's right here. Tuesday. Yeah, they seem to be more likely to be born on Tuesdays and maybe Fridays. We'll look more at the data here in this example. Okay, the hypotheses. The very general case of the hypotheses, to say it as simply as possible for you, for the null hypothesis, observed is equal to expected. The observed proportions, the observed counts are equal to the expected proportions, the expected counts. And the alternative hypothesis is that they're not equal. They're somehow different in some way. One of those P's is different from another P, is different from the expected count. Now there's two options. I know, two kinds of chi-squared tests. And for the first test, there's two options. These two options depend on your known or expected frequencies, what they are. So option one is the uniform hypothesis, when you expect all the probabilities to be equal, right? In this case of the births on the days of the week, there's no reason why you should think they would be different on one day of the week or the other. So you think they should all be equal to 1/7. In other words, your null hypothesis is that all probabilities are the same. Alternative hypothesis, all probabilities are not the same. For example, again, all hospital births are uniformly distributed in the week. P1 is equal to P2, et cetera, equal to 1/7. Option two is when you have some expected value to compare and your probabilities are equal to these expected value. That's what you want to test. Now, when would that possibly occur? Well, imagine that you cross homozygote parents and you look at these two co-dominant phenotypes, A and B. So in the F2 generation, and since you're all biology people, right? And a lot of you biology majors, you're familiar with this, right? Mendelian genetics. What would you expect? You would expect the probability of having AA is 1/4, probability of AB is 1/2, and probability of BB is 1/4, right? These are your expected frequencies. So if you wanted to know whether or not the parents of the in the F1 generation were homozygotes, you could test the genetics of the F2 generation to see if they follow these probabilities, and that would tell you, oh, look, they're the known the observed frequencies match the expected frequencies. You could find that out. And this would be one example where you'd say, okay, here, this is what I think is going on. And we're going to test against these numbers, not equal, but this particular group of proportions. All right, so probabilities are equal to expected values, probabilities not equal to expected values. The chi-square statistic. Chi-square. So again, this chi is like an X, sort of a wavy X. It's a Greek chi and written CHI, but pronounced chi. And this test statistic, again, helps to compare observed and expected counts or frequencies. The observed counts are the actual number of observations of each type, and the expected counts are the number of observations that we would expect to see if each type, if the null hypothesis were true. In other words, we would expect to see 100 babies born every day of the week if they were all equal and we had 700 births, 100, 100, 100, right? That is the expected count. And if that observed count is very different from the expected count, then we're going to get a really big chi-square test statistic. And a big chi-square, just like with all the other test statistics, a big t value meant a small p value, a big f value, small p value, a big chi-square value, small p value. A large difference of observed to expected, we're going to see that there's a significant difference. Working out this example a little bit more of the hospital births. So if our question is, are hospital births uniformly distributed in the week? And again, our null hypothesis here is that their probability of 1 is equal to the probability of 2. All the counts should all be equal to each other, equal to 1/7. The alternative hypothesis is that they are not all equal to 1/7. Something's different. One of these things is not like the other. And here we can make a little table and we can say, okay, on Sunday we had 84, Mondays we have 110, Tuesdays 124, and so on. Our total here again is 700, and our expected is 100 on each day of the week. Well, we can right away see, okay, there are fewer than expected on Sunday, and Monday there are the observed counts are more than expected, and Tuesday also more than expected. But is it enough to say that this is really a significant pattern? Is this a significant difference of the observed from expected? Well, here's how you do the calculation. It's actually really pretty simple, and it's a lot like the t-test. Remember, it was a lot like that, except before we were looking at the sample value minus the mean value divided by some measure of variance, right? In this case, instead of a measure of variance, we're just taking the observed minus the expected. That's kind of like the sample value minus the mean, and again, squared, divided by the expected, because we don't have a variance component here at all. There's no standard deviation. There's no standard error, nothing like that. So we divide by the expected. It's really, really simple. We actually just need those two values. Just that table that we had on the last slide will give us everything we need to do the chi-squares. And this is the code. I was just doing this in R before the class. I thought, you know what? Let me do this in R because previous lectures showed the results from a Minitab output. And since we're not using that anymore, I want you to be able to see what it looks like in R so I can help you to interpret it right from the beginning. So to do a chi-square test on these data, you just take 84, 110, 124, etc. And you say chi-square dot test, C for combine or concatenate. You put in all your numbers. And the default is that the proportions are equal. So R already just kind of goes in and says, oh, you've got 7 values. Each one should be 1/7. And it does it. And it gives you here a chi-square statistic, which here is 19.12. Your degrees of freedom is 6. Remember that we've been calculating degrees of freedom differently for different tests. But here, if the degrees of freedom are 6, can anybody guess how to calculate degrees of freedom for this test? How many days of the week did we have? I mean, how many days are there in the week? Come on, guys. 7, right? Okay, we all know this. Okay, so in this case, the degrees of freedom is the number of categories minus 1. So simple, K minus 1. And here our p-value for that degrees of freedom and this chi-square statistic is 0.003966, which is much less than 0.05. So we say, oh yeah, there is something going on. There is not a uniform distribution of births. Some of these days are different than others. And then if you have something significant going on, you might want to say, well, what? Why? Oh, okay, here's a little more about the chi-square distributions. So as with the other tests, the shape of the distribution depends on the degrees of freedom. The chi-square distribution generally is a strongly right skewed distribution. But again, it depends on your degrees of freedom. As your degrees of freedom go up, you get almost a more normal bell-shaped curve. And again, the degrees of freedom are calculated as the number of categories minus 1, or K minus 1. And here's how we could interpret our conclusions. We found that the number of births was not equal across all 7 days of the week. And here's how we always want to state our evidence, which is open parentheses. You put the test statistic equal to the test statistic, just copy and paste that from the R output, comma, degrees of freedom equals 6, comma, p-value. I just rounded here to 3 significant digits. I don't even think I rounded it right, but close enough. We get the idea. It's less than 0.05. So the next step is saying, well, how do they differ, right? And then you start looking at these percentages, these proportions, and you could say, well, 12% were on Sunday, 16% were on Monday, Tuesday had 18%, and look over here, Saturday only had 10%. So the weekends, right? This is how you could state your conclusions in terms of interpretation, the biological interpretation. The weekends, Saturday and Sunday, have the fewest births. Oops, should have put a parentheses here. Tuesday has nearly twice as many births as Saturday, 18% versus 10% of births, and 1.5 times as many births as Sunday with 12% of births. Well, why? And I don't know. And this is where you might want to ask some questions of the data or talk to some doctors. I wonder if doctors schedule births during the week. Remember, women don't always just go into labor just randomly. Some people have C-sections, and those C-sections are scheduled. And do doctors want to schedule it on a Saturday? Absolutely not. That's the last day they want to schedule it. If they can, they're going to schedule it for a Tuesday, a Monday or a Tuesday or a Wednesday. Fridays pop in there quite a bit, but definitely not Saturday. If something goes wrong, that probably means they'll also work on Sunday. So they want to schedule those births during the week. That's my guess. I really don't know, but looking at it, I would think that's probably what's going on. Does anybody have any other ideas? Does anyone know?
Maybe there's more stress during the middle of the week, and the stress hormones help induce labor.
Yeah, maybe. Yeah, it could be that. Could be that. Or maybe the mothers themselves are like, oh, I'd rather have like my baby born during the week because the weekends I want to have fun. I don't know. I don't know. Good. It's an interesting question. All right, now we're going to talk about the second chi-square test, which, like the one sample t-test and the two sample t-test, the one sample t-test isn't used all that often. The two sample t-test is the more useful test. And this chi-square test for association or independence, or the contingency test, multiple names, not my fault, just like that. This is the two-way test, and it's used quite a lot. Very, very handy. This is in chapter 22. If it helps you to remember which test is which, chapter 21 talks about the one-way test. Chapter 22 talks about the two-way test. So the chi-square test for two categorical variables. First, you start by making a two-way table. You construct your hypotheses for the two-way tables. You calculate your expected counts for the two-way table. You calculate the chi-square test statistic, and we'll go over the conditions for the chi-square test. And we'll talk about, oh, I'm not actually going to talk about the two sample z-test. I took that out. And then Simpson's paradox. Here's our example. Here's our example, because I think it helps to just walk you through an example. So smoking parents. High school students were asked whether they smoke and whether their parents smoke. Now here we have two factors, two categories, right? The first factor is the parent smoking status. That's in this first column on the left. The second factor is the student smoking status, whether or not the student smokes. Those are the row columns, right? So we've got the column and the row factors. And you see how this is now a two-way test. Before we just had one column or one row. Now we have a column and a row category. This is called a contingency table or a two-way table. The marginal distributions, I know this is a lot of jargon, but the marginal distributions are calculated in the margins of the table. In other words, over here on the right side and on the bottom, we calculate the totals. So we say, how many students are there in the larger group of students with parents that smoke? Whether or not the student smokes, do their parents smoke? How many total? 400 plus 1380 is 1780, right? And then how many do we have of students that smoke, whether or not the parents smoke? We have 1,004. If we add up all these totals, we get 5,375. That's our total sample size of all the students we asked that fit into all these different categories, right? Marginal distributions in the margins. The cells of the two-way table, each little box of the two-way table represents the intersection of a given level of one factor with a given level of the other factor, right? And this is how you calculate the conditional distributions. So how many students smoke where both parents also smoke? 400, right? And how many students smoke where one parent smokes? 416, right? These are your conditional distributions. And here's the total where both parents smoke. So the hypotheses for the two-way tables, a table with R rows and C columns, states that there is no association between the row and column variables in the table, which I think is a kind of wonky way of putting it. I think another way of saying it would be there's no association between the two factors, or to use this example, there's no association between students smoking and whether or not one or both parents smoke. Or the probability that a student smokes is independent of whether or not one or both parents smoke, right? It's not associated. They're independent of each other. Yeah, again, sometimes this is called the chi-square test for independence. And you compare the actual counts of the sample data with the expected counts given the null hypothesis of no relationship. Now, remember, for the one-way chi-square test, our expected counts were based on either the hypothesis of equal proportions or some known frequency. In this case, we're actually going to use the data to calculate what we expect the frequencies to be, and I'll show you how. Again, a two-way table has R rows and C columns, and the null hypothesis is that there's no association between the factors. So you calculate your expected count as the row total times the column total divided by the table total. The row total times the column total divided by the table total. Those are our marginal distributions, right? And the chi-squared statistic is summed over all the rows and columns in the cells. And it's the same math as for the one-way test, the observed count minus the expected count squared divided by the expected count. Same math, only here the expected count is figured out based on the data. Now, when you do have a significant chi-square, again, you want to spend a little time then looking at what's going on with your data. Well, what's different from what? And the largest components often indicate which conditions are most different from the null hypothesis. So you can compare the observed and expected counts or compare the computed proportions in a graph. You can also look at something called the contribution to chi-square, which we don't look at here, I realize, and I should calculate that for you during the lab. But let's say you're looking at the proportion of students that smoke when both parents smoke, right? And here you have that as 400 divided by 1780, right? There were 400 students that smoked and where both parents smoke. And there were 1,780 students where both parents smoke. And that means that 22.5% of the students smoke when both parents smoke. And here we have the probability of students that smoke where only one parent smokes, right? We had 416 students that smoked where only one parent smoked, and we had 2,239 students total where one parent smoked, whether or not the student smoked, right? That was our row column. And that's 18.6% of the population. And then there's a probability of a student who smokes where neither parent smokes is, again, 13.9%. So you can start to see when you look at these proportions, you can start to see the differences when you look at them on a bar graph. See, oh, look, the percent of students who smoke, given both parents smoking, looks a lot higher than when either one parent smokes or neither parent smokes. Now, again, this is kind of one of those situations where you say, okay, well, I really wish I could tell a little bit more about what was going on, because remember with the ANOVA, we were able to compare the groups. Specifically, we were able to say, well, are each of these really different from each other? Are they significantly different from each other? We can look at those confidence intervals. We can see if they overlap. We can do a Tukey's test and we can see what groups are different. There is a test for doing that with the chi-square, but we're not going to do it in this class. We're just going to look at the fact that, hey, there's something significant going on. And given these proportions, we think this is what's happening. All right. Okay, here's a bunch of numbers for you. There's a bunch of numbers. We're going to start by looking at what's over here on the right, the chi-square test. And we're looking at the expected counts are printed below observed counts. What is that mysterious legend telling us? Expected counts are printed below observed counts. Well, it is exactly what it says. So number of students that smoke versus number of students that don't smoke, where both parents smoke. So our observed data was 400 students that smoke where both parents smoke. But given the data, we would expect if there was nothing going on, if it was all evenly distributed, that given our total sample size, we would have 332.49 students smoke when both parents smoke. That's our expected count, right? And so on and so on. So here we have our observed count, expected. Here we have the observed number of students that don't smoke at all, 1,380. And if there was nothing going on, no association between these two factors, then the number we would expect to see here, given the total counts of all the data, would be 1,447, right? So here we see that the number of students that smoke that we observe is much higher than the number we would expect. And here we see that the number of students that don't smoke is much lower than the number we would expect if there were no association, if the null hypothesis were true, right? Those are some pretty big differences. Here we see our expected count is not that different from our observed count. 416 is our observed. We would expect 418. That's not a very big difference. Here again, we have 1,823 observed, 1,820 expected. Not a very big difference. Not a very big difference. Over here, where neither parent smokes, we observe 188 students that are smoking. And if there were no association under the null hypothesis, we would expect to see 253 students smoking, given all the students that smoke. But that number is actually a lot lower, which means there probably is something going on with having no parents smoking. You're less likely to smoke if neither one of your parents smoke. And if you don't smoke, let's see, our observed count is 1,168, and our expected is 1,102. So we actually have more than expected not smoking.
Last question.
Okay, which actually also makes sense, right? You're just less likely to smoke if your parents don't smoke. This is what it looks like, right? That's how I would interpret it. And this chi-squared test output is what you can get if you do a little bit more work, but when you're just running the basic analysis in R, if we look over here on the left, these big numbers here, this is what the R output looks like if you just plug in those numbers in a contingency table into R, as we'll do in the lab and as you'll do for your homework. And what you get here is your chi-squared statistic, which is 37.566. Your degrees of freedom is 2, and your p-value is less than 0.001. Very small.
I think we have a question.
Yeah.
So to find the expected counts, you do the single sample for one of the variables over the total for that variable for both samples.
I've got a worksheet. I've got a worksheet that we're going to go over after this lecture, and it walks you through it. Okay.
Okay.
Step by step. It'll be all super clear, I hope.